Thank you very much for that kind introduction, and to the Academy and the organizers for the invitation to present our work. It's an honor to be here and share our work with you, and thank you for being here. That was a tough act to follow, but I'll try my best to share some of our work. We became very interested in this with this one study we published now 8 years ago, in which we were looking at the modifiable determinants of obesity in young children. And this was a WIC population of toddlers that we had data to work on. Who was that? Somebody watching? Okay, Siri, go away. Okay, so we had about 1,500 children. We separated them into 3 groups based on their consumption of sugary beverages at 3 to 4 years of age. Never consumers, small consumers, or 2 or more servings per day. And there were 2 subsets. This first subset were children who were never breastfed or breastfed for less than 12 months. And then we looked at it was a very simple study where we looked at the prevalence of obesity, BMI above the 95th percentile in those children, and found this large association, which has been shown in other populations since then. But one of the more interesting findings from this paper was when we looked at these relationships, same separation into those 3 groups, but this time this group was children who were breastfed, not exclusively necessarily, but they were breastfed for longer than 12 months. So exactly the same relationship, but we found this remarkable protection from obesity, even in the face of the adversity of the high consumption of sugary beverages at 3 to 4 years of age. So we got very, very interested in trying to understand what it was about the role of not just breastfeeding, but extended breastfeeding that might offer this protection. So other studies have shown that sugar beverages are a risk for early obesity and other metabolic risk, not just in early life, but also during pregnancy. This is one such study. This is Project Viva, showing just very quickly in a large population that maternal consumption of high sugar in the 2nd trimester of pregnancy, not the 3rd trimester, was related to increased markers for obesity later in childhood. I think this was 7 years of age, and other studies have shown this as well. So there's certainly evidence now for a transmission effect of sugars during pregnancy, and data show that the earlier the introduction during childhood, the bigger the effect on obesity risk. We then became very interested in human milk oligosaccharides that were just introduced to you, and this work I've been able to do through my collaborator at UCSD, Dr. Lars Bode, who has helped with the analysis of the HMOs and the scientific direction of this particular study that I'll share with you. So this was previously discussed. I don't think this was particularly mentioned, but if you look at the major contents of breast milk, what's remarkable is that the oligosaccharide concentration is quite high, equivalent almost to the amount of protein in human breast milk, but it's quite unique to humans. In cow milk, for example, the oligosaccharide concentration is much lower. So we became very interested in the role of these compounds in obesity development, and later, as I'll show you, in brain development. But also we're interested not just in these oligosaccharides, but we became interested in simple sugars in breast milk like glucose and fructose. So just very quickly, this was already introduced. Human milk oligosaccharides are a combination of different products that are 3 or more saccharides in length, composed of different combinations of galactose and glucose. Lactose on the far left is not an oligosaccharide, it's a disaccharide, but you can see oligosaccharides build up from that backbone in various combinations and chain links based on whether they have fucose or other residues attached to them. So they're highly abundant, highly variable. I already mentioned in the previous talk, they're prebiotics digested in the infant gut, and apparently even earlier, before birth as well, there is exposure. That was new for me. Thank you very much for that, for your work. So just very quickly go through this. Different structural diversity, all the different oligosaccharides that exist in human milk. I'm just going to rapid fire through this because it was already covered. The path of HMOs in the digestion, or they're not absorbed. Wanted to share also this work, which Lars was also involved with, which is a collaboration with Megan Azad in Canada, which uses data from the Canadian child cohort, which is a remarkably rich birth cohort study, pregnancy cohort study, where they measured the variation of HMOs in over 400 women. And you can see the two groups on the right and the left that was mentioned. So there's the secretors who have a different profile, who make this one oligosaccharide, which is the most abundant in the secretors, 2FL. That's a genetic determination whether or not women are able to make this particular oligosaccharide. And then these are the non-secretors who are unable to make 2FL. So that's one thing. There's two subgroups, but within each individual, there's tremendous variation in the composition of the oligosaccharides. This is work in preparation. It's not published yet, but it shows the quite stable levels throughout the day. This was a study in which we gave the mother a high dose, in this particular case, of a sugary beverage at time 0, collected breast milk for 7 hours subsequently, and then looked at the variation either from day to day or hour to hour. We haven't published this yet, but you can see clearly from this data that levels are actually quite stable within women. Over time, there is some variation. This was all part of the Azad paper published in 2018. This is all cross-sectional. You have to have very good eyesight to see this, because what they're showing here is for each HMO cross-sectional data relative to time on the x-axis, which ranges from 10 to 30 days if you can't see it. But the point is, there is some variation, although I showed you within a day and from day to day. Over time, there is some variation in the content of these HMOs over time. And one thing we've kind of becoming interested in is 3SL up here, which increases over time, and 6SL, which decreases over time. So these are very, very similar structured oligosaccharides that have the role of delivering sialic acid for brain development to the infant. Some of them are going up and some of them are going down. So we don't yet quite understand how these oligosaccharides vary during the length of breastfeeding, which is of particular interest to us because of our finding of the role of extended breastfeeding beyond 12 months. We have a project underway looking at that, but I don't have the data to show you yet. Now, switching to sugars, just very quickly, I want to show you this metabolic pathway, which describes the fate of the predominant saccharides which are consumed, which are fructose and glucose. So in you and me, in adults who are fully developed, this is what happens when you consume. This would be sucrose, fructose connected to glucose, or if you're drinking a beverage with high fructose corn syrup, they already would be cleaved apart. But what's important to remember is that the fate of these two chemically identical molecules, only arranged differently in a pentagon here or a hexagon here, have very, very different metabolic fates. Glucose is absorbed in the gut, and it's used throughout the body for energy in every organ of the body, where it's combusted to carbon dioxide and water. Fructose is taken up almost exclusively by the liver at first pass. Estimates suggest in humans about 95% of fructose is taken up by the liver, which is non-insulin dependent, where it is converted to triglycerides. That is a metabolic fact. The triglycerides can then be stuck in liver and contribute to liver fat, or they can be repackaged and exported back into the circulation as VLDL triglyceride, producing things like uric acid and oxidative stress along the way. Now, if you consume more fructose in a fruit juice or in high fructose corn syrup beverage, you get more fructose going to the liver, more stress in the liver, more buildup of fat in the liver, and more buildup of lipids in the circulation. Now, if you're one of these, your situation is a bit different. If you're an infant, you were designed to thrive early on lactose, which is a combination of glucose and galactose. So obviously, there's a major difference here. There's no fructose in the natural feeding environment of an infant. So glucose is used for energy. Galactose is used to help brain development. Now, if fructose becomes inadvertently introduced into the diet, either through maternal transmission or through early introduction from the mother to the infant in juice or other nutritional products, infants actually aren't born with the machinery to absorb that fructose in the gut. You need GLUT5 to get fructose from the gut into the circulation. Infants don't develop that until there's enough fructose coming in nutritionally. So if you don't have GLUT5, that fructose gets stuck in the gut where it acts just like an HMO. It's not absorbed. It becomes a prebiotic and bacteria thrive on it, and it contributes to fructose malabsorption, which could have its own problems, as well as potentially gut microbiome evolution. Now, eventually, if there's enough fructose coming in, that's when you'll get the effect of fructose taken up by the liver, and the end stage of that is exactly the same as it would be in you and me, where fructose is metabolized in the liver. So to study all of this, we first did a short pilot study in 27 women. This was a collaboration with a former student of mine in Oklahoma, David Fields, where he had samples from a previous study in which we looked at HMO variation in breast milk samples relative to infant body fat measures at 1 and 6 months of age. The second study was an acute study in which we simply asked the question, if you give a lactating mother a high dose of high fructose corn syrup loaded with fructose and glucose, does any of the fructose and glucose get from the sugary beverage into the breast milk and therefore into the infant? Here's the main results from the first part of that study, where we simply looked at the composition of simple sugars in breast milk. And what we found was that there are detectable levels of fructose in human breast milk that are quite variable. You can see that the concentration is very low at micrograms per ml. So it's a very, very small concentration, but still detectable, and was significantly related not just to body fat composition, but actually to all measures of infant body composition, whether that was body weight, lean mass by DEXA, fat mass by DEXA, or even bone mineral content. So somehow here we have evidence for more fructose getting into breast milk being related to all aspects of infant growth. So where does that fructose come from? Well, the second study shed some light on that. This was the acute feeding study where we gave the mom at time 0 something that we shouldn't do, a large bottle of high fructose corn syrup loaded sugary beverage. On a different day, with the order randomly decided, they were given a similar volume of a beverage with non-caloric sweetener. And then we simply collected milk samples every hour for 6 hours and measured glucose concentration on this particular slide in treatment group. This was receiving the sugary beverage, and in red, the values of glucose in breast milk over the same time period after a non-caloric beverage. And what we see here is a small increase in glucose in breast milk by 2 hours, but it comes back down. The only time point that was significant was this one, and there was no significant difference in the total area under the curve. The picture for fructose was quite different. Same same beverage, same paradigm. Now we're looking at fructose in breast milk 6 hours after consumption. It rises much higher and it stays higher compared to the control group here. So fructose is elevated, sustained elevated up to 5 hours. Now you can see again, these concentrations are in micrograms per ml, and the question is, does it matter? Does a low concentration of fructose at micrograms per ml matter to an infant? And one study supports that the answer to that might be yes. This is not definitive because it's a different model. This is a study from a group at UCLA published in 2012 in which they looked at the effect of low concentrations of fructose on gene expression in preadipocytes. So fat cells are evolving. They evolve from a preadipocyte, and that's what infants are doing. They have fat cells, they're building fat, so preadipocytes are being developed into full-blown adipocytes. So this study said, well, what happens when you incubate preadipocytes with concentrations of fructose that vary from 0, 10 micrograms per ml, which conveniently was about the concentration we see in breast milk, up to 10 times higher or even 1,000 times higher. And they looked at gene expression and those 4 concentrations for PPAR gamma. And you can see for PPAR gamma, there was a significant increase in gene expression, but only when you hit 100 micrograms per ml. But for GLUT4, which is a very important modifier of overall cellular metabolism and adipogenesis and glucose intolerance, what was remarkable was that even at 10 micrograms per ml, which is the concentration we see in breast milk, there is a significant turning on of gene expression of this particular gene. So we need to think, when we're thinking about developmental effects, things can happen at very low concentrations that might have significant impact long-term on the infant. So we used some of these findings to design a larger study, which we're in the midst of now. We've fully recruited a sample of 240 Hispanic mothers. They're all subjects are Hispanic. We collect samples of milk and stool every 6 months. We measure body fat with QMR, echo MRI, and then we track the infants over time, looking at effects on body composition as well as cognitive development by 2 years of age. This just shows very quickly the composition of the cohort. It's equally recruited across the spectrum of maternal obesity, and they're predominantly vaginal deliveries and some C-section deliveries. The first study I'll show you, which is currently in revision, it's not published yet, but it's in revision, so it should be published pretty soon. But this just gives you an overall idea of the characteristics of the cohort shown here. Most of the women were secretors. There are some women who have this non-secretor phenotype and genotype. They're shown here, and you can see they're quite similar. Their age at delivery in years, BMI, and so on, shown down here. So the first study looked at the impact of different oligosaccharides on changes in body weight in the first 6 months of life. And what we found, looking at Lars's assay, looks at the 17 most abundant oligosaccharides in human milk. And of all those different oligosaccharides, we found evidence in this particular graph here of this one particular compound, LNFP2, which was the only one that was related to apparent protection from infant weight gain in the first 6 months of life. You can see women producing higher amounts of this oligosaccharide, their infants had lower weight change from 1 to 6 months. Now, we're also interested, there's multiple different mechanisms by which HMOs might impact upon obesity. One is through gut microbiome changes, which gut microbiome different gut bacteria may produce different products that may affect metabolism, brain development, energy regulation, and so on. And another is just that these HMOs act as we know, as prebiotics. So they may affect infant feeding behavior. So we're also monitoring infant feeding behavior through different, very hard to do in an infant. There's one questionnaire called the baby eating questionnaire. There's a child eating questionnaire. So at young ages, we can only use these questionnaires. When these infants turn into children, we can do more detailed tests, which we're planning to do on these children to test more definitively functional measures of eating behavior. But what we see here in another paper that is currently in review, we're looking to see whether any of these HMOs in milk, this is at 1 month of age, are related to food responsiveness, which is the drive to eat in an infant, which now, of course, it becomes very challenging because an infant is supposed to eat. So it might be a good thing that you have a big drive to eat in an infant, may not confer risk for future obesity, but studies show that as children get older, increased food responsiveness, drive to eat becomes a risk factor for obesity because they are less self-regulated and tend to eat more when given the opportunity. By 6 months of age, we can see now evidence for these 4 different oligosaccharides, which are significantly all related to increased food responsiveness at 6 months of age, increased drive to eat. That just summarizes some of the different findings. But this is just very early data from 1 month and 6 months of age, and we're currently getting more data on these infants. Now, as I also mentioned, we're also interested in how different HMOs might affect infant brain development, which is thought to be one of the main roles of these particular bioactive compounds. Animal studies show, for example, that this one particular HMO, 2FL, has a significant role for enhancing learning, memory, and attention in rodent models, and is already used by some formula companies as a supplement. So you can already buy some supplements that are enhanced with 2FL based on some of these studies, but there's actually no human studies that support evidence yet for any of these compounds in brain development and learning outcomes. So in our study, we're doing that by looking longitudinally at exposure to different HMOs during the first 2 years of life before a Bailey's test at 24 months of age to measure different learning outcomes. So the Bailey's test, if you're not familiar with it, determines outcomes at 3 levels: cognitive, language, and motor, based on different types of assessments. So cognitive tests, for example, in the 2-year-old involving placing shaped objects into a puzzle, or stacking. Gross motor tests, for example, measured by ability to step up and down these particular test steps. So what we found in this particular study, which will be in press, published in about 2 weeks in PLOS One, this is a study in which we looked at the determinants of, I can show here. But basically, it becomes a little complicated because you have all these co-varying outcomes. The main outcome is cognitive development at 24 months. And we look at different HMO concentrations. And of all the HMO concentrations at 1 or 6 months, the only significant factor that fell out of the relationship was 2FL concentration, but only at 1 month of infant feeding. No other oligosaccharides at 1 month or 6 months were significant in the model. Now also we have to take into account how much they received of the HMOs, and we do that by determination of how many milk feedings per day they have. So just looking at the number of milk feedings itself is significantly related to better cognitive outcomes at 24 months of age. But a lot of this variation is explained specifically by the HMO concentration in the breast milk. And here we see the single relationship between 2FL concentration at 1 month of age and infant cognitive development at 24 months of age, measured by the Bailey's score here. And you can see higher exposure to 2FL at 1 month of age resulted in a favorable outcome on the Bailey's test. Now, I'll just finish, even though the red light is flashing. I can stop or I shall continue. I'll continue. 9 seconds, because no talk on sugar is complete without low-calorie sweeteners, which is becoming very significant in our question of maternal and infant nutrition. So these are just some examples of different sweeteners. Splenda, fact of the day, sucralose is chlorinated sugar. This is just a sucrose molecule with 2 chlorine molecules tagged on. Makes it 600 times sweeter than sucrose, but not absorbed. And other sweeteners are shown here. And data, this is data from Alison Slavetsky, who works here at George Washington University, from Ann Haynes' study, showed that children who consume high amounts of low-calorie sweeteners actually end up consuming more calories and more sugar than those who tend to consume water. And the combination, the subgroup of children who consume both low-calorie sweetener and sugar-sweetened beverages actually can end up consuming 400 more calories per day and over 40 grams more sugar per day. So low-calorie sweeteners aren't doing the job they're supposed to do. Next slide, please. Thank you. And consumption is increasing in pregnant women. This is becoming very significant. This is just, again, Ann Haynes data from a collaboration with Alison, and the Ann Haynes data that shows increased consumption. Almost 25% of women now consume low-calorie sweeteners on a daily basis, mostly in beverages. And back to the Canadian study from Megan Azad's work, this was published in 2016, showing that women who tend to consume more low-calorie sweeteners during pregnancy, that's shown over here, higher consumers have infants with a twofold increased risk of obesity by 2 years of age. So there's a programming effect, and this finding has been shown in other studies as well, indicating that maternal consumption of these compounds may have long-term effects on the infant. And if you're wondering whether there's transmission of these sweeteners into breast milk, the answer is also yes. This is one study published by Rother in 2018 showing transmission of sucralose and ACE-K into breast milk. So I'd like to end there by thanking an amazing group of people that I'm blessed to work with who make all these studies possible and the outcomes possible and the papers possible and the funding as well. Thank you very much. I look forward to the discussion. Thank you very much. Thank you.